Hello, everybody, and thank you for joining us for another episode of Meet the Researcher. Today, I'm excited to welcome 2019 VHLA Pilot Research Grant recipient, Dr. Barbara Mysona. Dr. Mysona is an assistant research scientist in the Department of Cellular Biology and Anatomy of the Medical College of Georgia at Augusta University. Her research involves studying how the cells that make up hemangioblastomas alter blood vessel formation and why the blood vessels in hemangioblastomas behave in an abnormal fashion. Dr. Mysona, thank you for joining us.
Thanks, Josh. Thanks for that great introduction. As Josh said, I'm the 2019 VHL Alliance Pilot Award recipient. I wanted to thank the VHL Alliance for funding this work. It really means a lot to me, and I hope that the work I do can really make a difference for people with VHL disease. So as Josh mentioned, I work in Augusta, Georgia at Augusta University. And within Augusta University, we have the Medical College of Georgia. And this is just a little picture of our lab building. And of course, we're not in lab right now. We're working from home. And I apologize, we're starting a little late, had a little bit of a computer glitch. So hopefully everything will go smoothly. I thought I'd start by just telling you how I became interested in von Hippel-Lindau disease. So this week there have been great talks by the other award recipients. And like Dr. Magna, actually, I started out actually first as a chemical engineer. But then after working as a chemical engineer for several years and after having a family, I moved to biomedical research, and my PhD was in the eye, in the retina. And my whole life I had an intense interest in biomedical engineering and the function and the physiology of the human body. So I really enjoyed my PhD studies, and I have a background, unlike some of the other researchers, I don't have an extensive background in VHL disease, and I am not a clinician. I am a basic research scientist. So I started out studying the eye, and some of the things that I studied early on when I was getting my PhD turned out to be very relevant for VHL disease. So one of the areas I studied was diabetic retinopathy. And interesting, there's some similarities and some differences between von Hippel-Lindau disease, VHL disease, and diabetic retinopathy. But one thing that is common is an abnormal growth of new blood vessels. So for my PhD research, I worked a lot studying how glial cells in the retina interacted with endothelial cells. And I did a lot of what's called primary cell culture. So there's two ways to study various diseases. You can study with human tissues from people who have passed away, who donate their tissues. You can study in animal models, trying to look at the disease in vivo in an animal model, or you can do cell culture models. And primary cell culture models, the neat thing about them is you can dissect a problem. When you're in vivo, it's very hard to tease apart what do astrocytes do, what do endothelial cells do. But in primary cell culture, gives you the opportunity to look at the cells individually and then combine them together, see how their function changes. And so this primary cell culture background, I cultured primary neurons, I cultured primary Mueller cells, which are the main glial cell in the retina. And then once I got my PhD, I moved on to looking at endothelial cells in the diabetic retina and some of the factors in endothelial cells that change, that make them leaky. And then I moved on to yet another lab, which is the lab I'm currently in today. And I work with Dr. Catherine Bollinger, who's an ophthalmologist. And although Dr. Bollinger does not see VHL patients, she had a very interesting case of a young child who had another disorder called Sturge-Weber syndrome. And that syndrome also can produce angiomas in the retina. And so I had Dr. Bollinger is actually a glaucoma specialist, and I was working on astrocytes in the optic nerve, which the retina and the optic nerve are both part of the central nervous system. So, but our discussions about this young child with an angioma really re-sparked my interest in the vasculature. And by this time, I had been promoted to assistant research scientist. So that's a junior faculty, not a tenure track, but a faculty position. And it gave me the opportunity to apply for my own funding. So I was really looking for a project that I could call my own. And during this time, we're part of an institute called the Vision Discovery Institute. And through this VDI Institute, a piece of information passed over my desk about VHL disease. And I just became fascinated by VHL disease. It's a very unique disease. In that you have a set of genetic changes, genotypes, that are associated with phenotypes that are part of the disease. So I combine these two interests, the interest in primary cell culture, actually maybe three interests, the interest in astrocytes and glial cells, and the interest in abnormal growth of new blood vessels to study VHL disease. And you might be asking me, well, Barbara, you started out in the retina. The title of this talk is The Role of Astrocytes in the Brain Astrocytes in VHL disease. Well, the retina and eye are a very difficult tissue to work in because if you're using an animal model like a mouse, the mouse eye is just so tiny. You really can't get very much tissue out of the eye. It's a very, very challenging. So that's why that one of the things that fascinated me about von Hippel-Lindau disease is there are similarities in the hemangioblastomas that form in the retina and those that form in the brain. So I made the jump from the retina to the brain, but being part of the central nervous system, as the talk goes on, there's a lot of similarities in the organization of the cells that support the blood vessels, both in the brain and in the retina. So I always like to go back and talk about or look at history, because I think we can learn a lot by looking at what people have done before us. And there's been so much work that's gone into studying VHL disease. So and many of you who are VHL patients probably are very familiar with some of this, but it's always good, I think, to reflect on the history of a disease. So Eugene von Hippel in 1904, there were some other people that noticed angiomas in the eye, but he was an ophthalmologist, which of course struck a chord with me because I had an interest in the eye. And he recognized that there were hereditary angiomas in the eye and that these were abnormal growths that had new blood vessel formation. And then in 1927, Arvid Lindau, a Swedish pathologist, he recognized an association between the angiomas in the retina and the hemangioblastomas of cerebellum. And histologically, the angiomas in the retina are similar to the hemangioblastomas of the cerebellum. And so the term von Hippel-Lindau disease came into use in 1936, but it really wasn't until 1964 that we had a diagnostic criteria for VHL disease. Really, the big breakthrough for VHL disease that's allowed so much work to go on is the location of the VHL gene to the short arm of chromosome 3. And in people, we have 23 pairs of chromosomes, and by now, all the genes that produce proteins anyways, have been mapped by location to these 26 chromosomes. So on the short arm of, first, just to discover that on the short arm of chromosome 3, there was the VHL gene was a big discovery. But shortly after that, only 5 years later, the code, the genetic code of the VHL tumor suppressor gene was defined. And that let scientists know that the VHL protein is a small 221 amino acid protein. Proteins are made out are actually polymers made up of chains of amino acids. And it turns out this gene can code two different sizes of the protein, a 30 kD protein and a 19 kD protein. And kD stands for kilodalton. It's the size of the protein. But to give you a feel, large protein would be 160, 170, 210 kilodaltons, and a medium protein probably 60 to 90 kilodaltons. So the VHL protein is small, but once the protein structure and the protein amino acid code was discovered, it let people clone the protein and also knock out the protein. So scientists from 1990s to today have discovered many VHL protein functions. And in my slides, PVHL refers to the protein. And so on this slide, it's a little bit of a complicated slide, but it does not highlight all the VHL functions, but the ones that are really relevant to my project. So the VHL protein, it kind of has two main arms or two main pathways through which it exerts its effects. One is through the regulation of a protein called HIF. And HIF is a hypoxia-inducible factor. And one of the key things about the VHL protein is when you have normal protein that's not mutant, the cells, when they're in their normal environment with the normal amount of oxygen, we call it normoxia, the HIF has two subunits, an alpha and a beta, and the alpha subunit is the one that is really kind of regulated by the VHL protein. So in normoxia, HIF alpha levels are low. And they're low because the VHL protein, in combination with some other proteins, works to dispose of the HIF alpha, sends it to the proteasome, which destroys unwanted proteins. It's like the garbage disposal. So but when there's hypoxia, there's various mechanisms by which HIF alpha no longer binds to VHL. And so then HIF alpha with HIF beta is able to combine and go to the nucleus, where HIF alpha activates a whole slew of genes that are required if you have hypoxia, low oxygen. So the VHL protein has a very important function, and in terms of helping the cell to behave properly when there's plenty of oxygen, or to respond to hypoxia, which is a life-threatening insult to the cell. And when you have a mutation in the VHL protein, the regulation of VHL, the taking HIF alpha out to the trash, basically, is disrupted. So then in normoxia, HIF alpha tends to accumulate. The HIF genes are activated. They shouldn't be activated because there's plenty of oxygen, but they're activated anyways. And so what are these things that HIF activates? A lot of them are growth factors like VEGF, PDGF beta, TGF alpha, erythropoietin, and these factors are all designed to bring more oxygen to not just the cell, but probably the organ or the tissue the cell resides in. And so how does an organ or a tissue, the brain, the retina, the kidney, how do they get more oxygen? They need more blood vessels. So a lot of these factors are associated with blood vessel growth that eventually results in tumor formation. So the boxes on the left, highlighted in red, show the results of work of numerous scientists, and we're still trying to understand how a defect in the VHL protein leads to this tumor formation. And if it's any solace, of course, to people with VHL disease, it's a very trying condition. But if there's any silver lining to this disease, is that the basic function of VHL protein is involved not just in VHL disease, but many types of tumor formation. So the more we understand about VHL protein will not only help VHL patients, but other cancer patients as well. So on the left-hand side is the HIF regulation. The other arm is much less well studied, and those are other actions of VHL protein. So these include healthy VHL can bind to another transcription factor called SP1. And that, interestingly, can also be associated with a reduction of VEGF levels. VEGF is one of the key proteins involved in stimulating new blood vessel formation, whether it be in diabetic retinopathy or VHL disease or hypoxia. VEGF, it plays a big role in blood vessel formation. One really interesting action of VHL protein under other actions is that with normal VHL protein, it will bind to another protein called fibronectin that's a key part of the extracellular matrix. What is the extracellular matrix? It's basically a whole group of proteins that are deposited outside the cell. But especially for blood vessels, we call it ECM. the formation of this matrix, which you think is like a web of proteins, and they help support the cell. They signal to the cell. And for blood vessels, the ECM plays a big role in having a proper, normal, healthy blood vessel formation. So it's very interesting that the other actions of VHL, particularly in regards to fibronectin and the assembly of the extracellular matrix, in the VHL protein, that other arm of actions are also disrupted. So this abnormal ECM formation may also play a big role in tumor formation. And so really this basic molecular biology and function of the VHL protein all links back to the kind of tumors that patients with VHL experience. And as many of you who have VHL disease know, it's inherited, it's autosomal dominant, meaning if you have a parent who has it, approximately 50% of the children will have it. It's approximately 1 in 36,000 to maybe 1 in 50,000 live births. The tumors can be benign or malignant. And, you know, that's one of the big mysteries surrounding VHL protein and VHL disease, is why some tumors are benign, why some are malignant, and the tissue specificity of the disease. Why does VHL disease affect the brain, the cerebellum, the spinal cord, the retina, the kidney, and the adrenal glands most commonly? There's some other areas like the ear that are affected. But why does this defect affect those tissues specifically is a really big mystery, I think, in the function of the VHL protein and VHL disease. And these tumors occur in young adulthood, so in the 20, 30, 40, 50 year age range, although they can occur throughout life. So these hemangioblastomas are what I'm really interested in. They are a hallmark of VHL. They occur in 60 to 84% of the cases. The good news, they're typically non-cancerous. Often the retinal hemangioblastomas are the first sign of the disease. And Dr. Matnya gave a great talk about the retina the other day. And but it also affects the cerebellum, the brainstem, and the spinal cord. And although these hemangioblastomas can be removed, they're in a very sensitive part of the nervous system and can cause really serious or life-threatening complications. So currently, our interventions include the retina, laser photocoagulation, careful timing of surgery, but the non-surgical options currently are fairly limited. We have some really, promising studies on chemotherapy drugs that act to decrease the vascularization of tumors. There's also been some work trying VEGF inhibitors, but those drugs don't work as effectively as we might hope in VHL disease. So hopefully my project will help us understand better how these hemangioblastomas in the cerebellum are formed, if we can understand that, maybe we can find some better therapeutic targets, targets that will help specifically stop the formation of hemangioblastomas in a VHL disease. So the cerebellum is a very interesting part of the brain. I actually have a personal connection to this part of the brain. My dad, a year and a half ago, had a very bad cerebellar stroke. And so I'm quite familiar with some of the neurological actions of the cerebellum. But one thing, it's an area of the brain kind of located low and in the back. It helps provide smooth and coordinated movement. And the first picture shows kind of, we call it a sagittal view, a longitudinal view of the brain. And if we were to blow this up and look at the tumor that forms in the cerebellum, the red part of the tumor is the solid kind of mass of the tumor. But because the tumor has all these leaky vessels, often fluid can build up around the tumor. So that's a big problem because the fluid puts pressure on the rest of the cerebellum and can cause headaches, nausea, a wide variety of symptoms. So really, I'm really interested in understanding the composition of the hemangioblastomas and what role this cell that I'm interested in, the astrocytes, play. So if we look at the hemangioblastoma in this cartoon at the bottom, it shows some of the cells that are involved in the hemangioblastoma. So one is these very abnormal leaky capillaries. They're made of endothelial cells. We often abbreviate it by the term EC. So the endothelial cells are the cells that make up the blood vessels. There's another cell called the periocyte, which is P in this cartoon. And periocytes are interesting. They're associated with the capillaries, and they have a role of supporting the capillaries. Then there are stromal cells that lack the functional VHL protein. And these are the cells, the studies on these hemangioblastomas, the origin of these cells is not quite certain, but they seem to be, if you want to call them, the cancerous cells. They don't have functional VHL protein. There's a cell called the mast cell, which is an immune cell that signals between the endothelial cells and the astrocytes. And then there are the astrocytes. Now, astrocytes tend to form a lining around the edge of the cyst. And the astrocytes have they're not round cells. They have a very raggedy appearance with long fingers that wrap around and touch blood vessels. They touch neurons, and astrocytes play a very supportive role in maintaining the health of the neural tissue. So that's part of the reason I have such an interest in the astrocytes. So how are astrocyte endothelial cell interactions altered in VHL disease? And that's really my interest. What's going on in the communication between the astrocytes and the endothelial cells that may occur early on in the disease? We know later in the disease, they're located around the periphery of the hemangioblastoma. but what is their action? And astrocytes, I know from my experience in the retina that the astrocytes play a big role in regulating the endothelial cells. So this cartoon from Sam Ajasim at the University of Portsmouth is a beautiful illustration of the blood-brain barrier. And this barrier, when you have leaky vessels in a hemangioblastoma, the blood-brain barrier is compromised, which is probably part of the reason the glial cells, the astrocytes, try and form a boundary between the hemangioblastoma and the rest of the brain. But you can see in this cartoon that first, kind of the inside core, you have the endothelial cells, then you have the periocytes, but then you have the astrocytes, and you also have a role of the microglia, the macrophages and the mast cells, and really something that I have not read much about at all in terms of VHL disease, the neurons. And so all these different cell types interact to form the blood-brain barrier. So our hypothesis in this study of the role of astrocytes in VHL disease is that we hypothesize that VHL mutations in astrocytes are capable of inducing the malformed blood vessels independent of VHL mutations in endothelial cells themselves. And in forming a hypothesis for a project, you have to take a stand. So this is the stand that we took, and we will test this hypothesis to see if it's true. So our first goal was to isolate and characterize primary cultures of these cerebellar astrocytes and endothelial cells with and without VHL protein. And our second goal is to determine how the VHL protein in astrocytes, really the mutant VHL protein in astrocytes, affects endothelial cell function, but there may be some things we learn about how normal VHL protein in astrocytes affect endothelial cell functions as well. So our experimental plan was really pretty simple. This is the plan that's in progress. So we get VHL wild type and mutant mice. We isolate primary cells from the cerebellum with and without VHL protein. And we're going to look at astrocytes and endothelial cells with the VHL defect compared to normal VHL separately and then together in co-culture, because there's a lot of communication between astrocytes and endothelial cells, and we suspect that astrocytes and endothelial cells may behave very differently when they're apart than when they're together. So the first step in this plan is you need a mouse model. And I thought that Dr. Matnya yesterday did a very nice job of explaining some of the challenges we have in VHL disease. So in a sporadic cancer, you would need 2 VHL mutations to lead to tumor formations. But in VHL disease, which is a hereditary cancer, you have one inherited allele, and an allele is that one part of chromosome 3 pair, one that has the defect, and then the other, you get an acquired mutation that's occurs sporadically, and that leads to the tumor formation. And, you know, in many diseases, if the protein defect is not embryonically lethal, you can just have a knockout mouse that's very easy to handle and genotype. But in VHL disease, it's very, very interesting that homozygous VHL disease is lethal, both in people and in mice. But the heterozygous VHL defect, people that are heterozygous survive. So we need a little bit of a fancy mouse model. And mouse models have really developed over the years. And we now have a system called an inducible Cre-Lox VHL minus indicates the mutant VHL model. And so this mouse model, I believe, is a little bit slightly different than Dr. Matnia's, but we're lucky in the Jackson Labs. That's a lab that specializes in mutant mice, transgenic mice, and we can purchase 2 different mice from them. And I'm using 2 mice, one called the CAG-Cre mice and one the VHL TM1JAE mouse. And we can combine those mice to produce an inducible Cre-Lox VHL model. So what does that mean? So these inducible Cre models allow us to manipulate the expression of our desired protein in both space and time. So, you know, depending what Cre mouse you choose, you can express a protein called the Cre protein just in the liver, just in the eyes, or you can produce it throughout the whole mouse. And our strategy in this project was to combine a CAG-Cre mice, which essentially produces Cre throughout the mouse. And that mouse is viable, it's healthy, it's a good mouse to serve as the control mouse because you would like to know what Cre protein does it by itself. And we cross that mouse with the LOX-P mouse, which is the VHL TM1JAE mouse. And so this mouse, the VHL mouse, it has the promoter and X1 are flanked by a sequence of DNA base pairs, and they're called LOX-P. The LOX-P protein is in bacteria. It's not in people. So there's no homology to any human proteins. But these transgenic mouse with the FLOX gene, this lock with the LOX-P site, it's the target of the Cre protein. So in this model, it's quite a neat trick. In the CAG-Cre mice, the Cre protein is sequestered. Well, suffice it to say, we have a very nice mouse model that this funding for my pilot project allowed us to purchase these mice, to cross them. We crossed them several times. And just about the time we were sent home because of COVID-19, we were really ready to produce some really interesting mice for studying the VHL disease. So with these mice, we can culture primary astrocytes. We euthanize the mice humanely. Once they're passed, we remove the brain. We add some, we cut it up, we digest it with an enzyme called trypsin, and we can actually plate this tissue on a coated flask. And these astrocytes, one of the proteins that's characteristic of them is called GFAP. It's here we use an antibody to identify the protein in the cell. It lights up green. So on the left, you can see how the astrocytes look under the light microscope. But they're a very flat cell with kind of long arms. They're much easier to view with an antibody that will tag that particular cell. and light it up. Similarly, we can culture the primary endothelial cells from the brain. That's a little trickier. We have to insert a step to separate the microvessels from myelin. Myelin is the lipid-rich material that oligodendrocytes secrete to surround axons. But basically, once we remove the myelin, we can digest the microvessels. We actually use some magnetic beads to pull the cells out that are positive for another protein called CD31 that's unique to endothelial cells. And we can get a pretty nice culture of endothelial cells. In the light microscope on the left, they kind of have a cobblestone appearance, but on the right, you can see the red is the CD31 that's on the cell membrane. So once we have the astrocytes and endothelial cells, this part of the work is kind of where we got cut off with the COVID-19. We can grow these cells apart, so we can get astrocytes with and without with the VHL protein or without VHL protein. We can get endothelial cells with and without the VHL protein. And we can look at some of those proteins, HIF-alpha, VEGF receptors, PDGF-beta, fibronectin, and we can start to tease apart what astrocytes do, how they behave with and without VHL protein, how endothelial cells behave with and without VHL protein, and we have a pretty nice system using a special cell culture dish called a transwell plate, where we can grow astrocytes on the top and endothelial cells on the bottom, because astrocytes secrete a lot of factors that affect endothelial cells. And likewise, endothelial cells in trouble without VHL protein may be secreting factors that stimulate the astrocytes. So we can look at 4 different combinations, normal endothelial cells and astrocytes, abnormal endothelial cells and astrocytes. And then we can break that analysis down further and see whether it's the VHL mutant or lack of VHL astrocytes that's affecting endothelial cells or vice versa. And we have another nice assay that we like, which is the tube formation assay. And that's specifically for endothelial cells to see if they have the tendency or an enhanced tendency to form new vessels. So that's kind of where we are at currently. Our primary astrocyte and endothelial cell cultures are working. We're waiting on mouse breeding to get our final and really enough mice of the special genotype that we're looking at. And in the meantime, we're going to look at some hypoxia and normoxia experiments in wild type or normal mice. And just so we can understand normal astrocyte endothelial cell interactions and perfect our techniques and conditions. And really, although this is a work in progress, the completion of this work is expected to give us the foundation for applying for additional NIH funding, National Institute of Health funding, to submit some peer-reviewed publications on our findings. And the significance for VHL disease the hope is that this is going to provide new insights on the mechanisms of hemangioblastoma formation and the role of astrocytes in the cerebellar astrocytes in this process. And hopefully it'll help us to identify some new targets for VHL disease. And hopefully that's something to give hope to VHL patients, although, we call it from the bench to the bedside. hopefully the work we do as basic scientists at the lab bench will help lead to a development of a treatment at the bedside. So an understanding of VHL disease will benefit all cancer patients, because this tendency of tumors to kind of hijack cell machinery can be linked to the VHL protein and the cell thinking that it's hypoxic when it really isn't. So I just want to once again say a very sincere thank you. Thank you for listening today, and thank you for all your support of VHL Alliance and the opportunity it's given me to participate. And I enjoyed talking with everyone today, and I'm glad to try and answer any questions.
Dr. Mysona, thank you so much. We have a few questions for you. This sort of goes off of how you concluded the presentation, but why should people outside of the VHL community care about and support your research?
Oh, I think that's a great question. Well, I think it really, the VHL disease, although the disease itself affects a relatively small number of people, when you think of how cancer, all the different cancers, breast cancer, prostate cancer, lung cancer, I mean, we have a huge number of cancers where the understanding of how first different cell types interact, and also understanding how tumors form, it's really something that's important to all tumor research. And if we can understand how a VHL protein and its abnormal actions contribute to the cell thinking it's hypoxic and it's not, we can really understand why there's blood vessel, abnormal blood vessel formation in a lot of tumors, because a tumor is basically a hypoxic environment. So how does a tumor start? What's going on in the cells of the cancer cells that's tricking the normal tissue into thinking that it needs to grow blood vessels to support the tumor? Because if a tumor didn't have any blood vessels, it wouldn't survive. And I also think VHL disease may have some very important insights for other diseases such as diabetic retinopathy, because there's a lot we don't understand in diabetic retinopathy between the interactions between the astrocytes and the endothelial cells, and how blood vessel formation goes awry. So I think is really, I think that's one of the things that attracted me to VHL disease. Although it's a mutation that affects, it's considered a rare cancer, it really understanding the VHL protein and its actions has huge potential for us understanding a lot of vascular diseases.
Thank you. A couple more questions. Do you think the coronavirus pandemic will have a long-term impact on your research?
Oh, that also is a good question. In the short term, it's disrupted our research, but it's given me time to do this Facebook Live, which really, when researchers have to sit down and revisit their ideas and present them, it really helps them to gain a better understanding of their own work and why it's relevant. So in that sense, even though we've been away from the lab, the time has been well spent. I think I'm in Georgia. I think we'll get back to the lab pretty shortly. So, but what I'm afraid of is the economic impact that the COVID-19 has caused. I think in the coming years, it may affect the research funding. And I think that will be the greater effect on the research, because all our budgets are good through, all our government and state budgets are good through the 1st of July. But then different funding agencies are going to have to look at what their new budget is. And that probably will slow things down. But when things are slow at the bench due to shortages of funding, usually it frees up more time for thought. So hopefully, I like to try and be optimistic. It's not good, but hopefully good will come from it.
Yes, I agree. Thank you. Here's another question for you that sort of goes off of that is, what do you think the VHL research landscape will look like in the next 5 to 10 years?
Oh, that's good. I'm hoping, my hope would be that we start to learn more about the whole process by which you have a heterozygous mutation. So you have one good allele with a good VHL protein and one bad allele with the mutant protein. So my hope would be that we really start to understand the process by which the healthy allele, the good allele, either goes under some random somatic mutation, or perhaps the good allele is being silenced. We know there are these epigenetic modifications that can mask the expression of a gene, even though it's there. So I think that area of research, kind of going back to the earlier question, why is VHL research relevant to somebody who doesn't have VHL? Well, all cancers have this basic characteristic that certain genes that are normally expressed are now masked. So I think understanding that, I think in addition to the good work that's going on in so many areas, I think understanding that question is really key to understanding VHL disease and understanding cancer, because if you could prevent the good allele from going bad, then you could possibly, postpone the various types of tumors that occur in VHL disease to much later in life.
Thank you. And one final question for you. For a person who is clinically diagnosed with VHL, however, they have an inconclusive genetic test, how do if they had a daughter who also had a genetic test, that was a negative, how do they determine if continued screening is warranted for both of them?
That's a good question. We have so much to understand about the penetrance of these diseases. Meaning, if you have a disease, how does it manifest itself? And so there are many different VHL disease, very interesting. So the genotype and the phenotype are linked, but there are probably many VHL mutations that are still to be discovered. So maybe you're screened, that test doesn't show a mutation, or maybe you have a mutation not in the VHL disease, VHL protein itself, but maybe some related protein that interacts with the VHL protein. And in that case, a screen for abnormal VHL protein would not detect that. So I think it's best, if to keep to keep track in terms of various symptoms, what's going on. Because sometimes in medicine, the treatment is the same whether you know whether you have the disease or not. So you know that these, probably regular eye exams to make sure, since hemangioblastomas in the retina are one of the first signs. And mind you, I'm not a clinician. I'm just speaking here as a basic scientist. Probably monitoring your health is a good idea. I actually have the symptoms of a disorder called Marfan's disease. I have not been genetically specifically tested for it, but the treatment is still the same. That disease can affect your heart, your aorta. So I get screened every 2 years for it. So probably, basic screening is a good idea. But considering that VHL disease, the symptoms can be be very bad in one person and not so bad in another. So just live your life and enjoy it and enjoy every day you've been given. And that's about all I think any of us can do.
Dr. Mysona, thank you so much for taking the time to join us today, for sharing some of your research, as well as for all the important work that you're doing. We really appreciate it.
Yeah, thank you. And I'm sorry for the various computer glitches.
No, it's okay.
COVID-19 era, we're all getting better at this stuff.
Yep. Thank you so much.
Thank you.
For everybody watching at home, you could find a calendar with upcoming Surviving and Thriving Together series events, as well as recording of past events at vhl.org/together. We hope you'll join us again. And until then, please stay safe.